if it is like 3 minutes, like maybe I can just make a ding or something if you're kind of like going over time or something. So I'll let you know if it's like maybe like 3 minutes, like 1 minute, especially if it's 1 minute left. I'll chime in so that we can't have people run over time. So, all right, thank you.
We'll mute.
Yep, let's mute.
Should we leave our videos on or off?
I think you can leave it off for now, but when you're presenting, you can put it on.
Okay, perfect.
Should we go ahead and get started?
Yes. So let's get started. Second. All right. Hello, everybody. I would like to welcome you all to this session titled Direct Collaborations Between Model Organism Researchers and Clinicians. I'm Shinya Yamamoto from Baylor College of Medicine, who will be chairing this session together with Andy Golden from the NIH. Andy, Koichi Kawakami, Philip Campo, and I initially proposed this session as a workshop for this meeting. Thanks to the organizing committee, we were fortunate to have this upgraded as a thematic session because the topic seems to be interest of many people in your community. In addition to the interesting science that are being studied, we hope the talks will also inform you why model organism is important for clinical research and how one can work with clinicians in a productive manner to make direct impact on patient care, ranging from disease diagnosis to therapeutics. The first speaker is Ada Hamosh, who is a human geneticist, and she'll tell us about the essential roles of model organisms for functional studies of genes and variants linked to human diseases. Ada?
Just my screen. Can you all see a screen?
No, not yet.
This is very odd. Hang on. There you go. Okay. I'm sorry. 2 buttons I was missing. Excuse me. So my name is Ada Hamosh, and I'm an actual clinician. Researcher. So I know that not all of you may know what clinicians do and how we start to look for model organism researchers. So what do? We evaluate a patient, and we try to determine if recognize. We do that in a second. And then we send the indicated test, which may begin or may end with a trio X sequencing, whose results may be an answer, a known variant, known disease gene, which matches the patient's phenotype, and we're done. Another possibility is no answer at all. I don't see any variants. The lab says we see no variants that might be implicated in disease. Okay, well, we don't know what to do with that scenario, but that is an outcome. But sometimes we get a more recessive variant or variants in a gene not yet implicated in disease. And then we need to figure out, could that actually be real? And I'll tell you what we do to do that, but then we really need you. And sometimes we find a variant of uncertain significance. Yes?
Oh, sorry. Yeah, your sound is a kind of like crippling. So maybe can you turn off your video to make sure we have good bandwidth? Thank you.
My video, like I don't know how to do that when I'm in this mode. I'm sorry. Let's see. Oh, there we go. Can I? Sorry. Go back. But sometimes we find a missense variant in a gene of known function, but we don't know the role of that variant. So again, model organism researchers can be very helpful in understanding that. Finally, even if we know the gene, we know the variant, we know the disease, we need to figure out how to treat it. And to understand the biology, we really almost always need model organisms. And depending upon the kind of phenotype, depends on which model organism a clinician might want to work with. So the first thing I told you is we try to determine if what the patient in front of us has is a 18 years, so I am biased. Anyway, it is a catalog of human genes and genetic disorders, and each entry is a stable number. There are titles and aliases. It's a rich, full-text description of genes and genetic disorders, including structure, function, animal models, clinical features of the patient's pathogenesis, and clinical synopses. It's expert curated and has a predictable structure, and it's cited to the peer-reviewed biomedical literature. That's the structure of the database. So there's gene entries and phenotype entries. They're linked in a gene map. There are allelic variants, but we are not the variant registry. As of 406 entries, of which 6,300 almost are exactions, and about 8,000 are phenotypes. Some of them are distinct. Some of them are most example, type 2 diabetes. These are the data through the end of 2019, and we are in a steadily growing phase. There's no evidence of slowdown of discovering new gene-phene relationships. This is just to tell you what a gene entry looks like if you're not familiar with OMIM. There's a lot of useful information just at the very top of the page. You can see the clinical synopsis associated with a particular gene by looking, they can look at it side by side and see what the similarities or differences are. You can look at phene gene graphics, or a visual way that we've shown people how, oh, I'm sorry, I went to the example variants here. You can go to ClinVar variants to get more, and you'll hear more about ClinVar a little later from Shinya. You can, the green box shows a visual representation of the relationship between genes and phenotypes in OMIM. This is a PDF that explains what these relationships and all these symbols mean, but this is what it will actually look like, and each of these things are hot links that'll let you know more about the disease, the gene, and their relationships. You can also, that's a linear view, that's a radial view, and it rotates in real time, but I'm sorry I did not capture it in that way. This is a phenotype entry. This is one of the phenotypes caused by the gene I just showed, structured appropriately for a phenotype, so not exactly the same information as what's in a gene. We have clinical synopses, phenotypic series, which shows that there's more than one gene that can cause Sickle syndrome, and they have different numbers to distinguish them. And then we show the same diseases if you're looking a bust, and it'll give you a picture feature. So this girl has, yeah. We all have all the feature IDs from all the resources you could possibly want, and all of this is available from the back through the back end with a robust API. And you can see them side by side, and I don't know what else I'm showing you. We have copious links to everything else. So if you look up a gene, you can stumble, yep.
You can call in because your sound fades away and then comes back. And
oh, I'm so sorry.
you've
So-
been given a phone number, and so you should use that phone number.
So actually, why don't we actually move on to like so let's come back to Ada's talk. Maybe like we can do Hugo's first, and then then we can actually try to get the technical stuff corrected. Is that okay?
Let me start.
Yeah.
That's fine. I can start.
Yeah. So what? Yeah. Why don't we get Hugo's talk first, and then we'll come back to Ada later on.
Okay.
Sorry about that.
I need to be able to share my screen.
Yes.
Right?
Now you can share your screen, please. Sorry about this technical glitch.
Can you see my screen?
Yes.
All right, let me know if there is a sound issue. So we're a part of the Model Organisms Screening Center of the Undiagnosed Diseases Network, and our goal is to help in the diagnosis and the molecular pathogenesis of genes that are being discovered in rare disease patients. And so the Model Organism Screening Center consists of 3 people, myself, Michael Wangler, and Shinya Yamamoto, and a zebrafish team, Monty Westerfield and John Postlethwaite at the University of Oregon. I will not be talking about the zebrafish much and focus on what we do at Baylor College of Medicine in the Model Organism Screening Center. The whole story started about 10 years ago when we embarked on a screen to identify homozygous lethal mutations in fruit flies and a screen for progressive degeneration, where we generated in the heterozygous flies that are shown at the bottom here, clones, and then shine light and recorded electroretinograms in young and old flies. And by doing so, we were able to identify numerous mutations in numerous genes. We also screened for a variety of other phenotypes like bristle loss. And eventually, once we had identified a robust phenotype, we'd perform electron microscopy on most of these. This allowed us to identify 700 mutations in 165 genes that are listed in this slide. And as you can see, on the left are the fly genes, and then next to the fly genes are the human genes. And what we noticed first is that most of the genes that we identified at human homologue, only the ones that are shown in gray here, are fly genes that don't have human homologues. So that's only 12 out of 165. We also noticed that many of these yellow genes were already annotated in the OMIM database, and that at the time, 30% of the genes had human homologues that were identified in the OMIM database. Now this is 61%, and all the genes that are shown in red were discovered since we published this paper in 2014. So this obviously brought us in contact with lots of physicians and human geneticists and allowed us to discover a set of new human disease genes and prompted us to become active players in this community. And we quickly realized that model organisms could have a much more important contribution than they had essentially provided in the past, in the sense that we could participate in the diagnosis. And one of the major reasons is that the profile of human genetics has changed in many cases now. We only sequence one individual, as is shown here, this kid and the 2 parents. Numerous mutations or de novo mutations are only found in the patient. And this is an N of 1 situation where when you look and analyze the genome, you typically come up with a number of variants that are possible candidates for the gene, but you have to show which variant is really responsible. And human geneticists are pretty good at coming up with ideas and propose genes. And we can then use model organisms like flies or zebrafish. We've now also had a C. elegans team, or mice, to help in the diagnosis. And then, as Ada was mentioning, work out the mechanism and use that to screen for therapeutic targets. So we became part of the Undiagnosed Diseases Network, which, can consists of a lot of clinical sites, now 12 clinical sites. The UDN has expanded. And the patients can apply to the coordinating center of the Undiagnosed Diseases Network and get assigned one of those clinical sites that are in any of these states with red boxes. And typically they get a workup, a phenotypic workup, and once this phenotypic workup has taken place, they move on to 3 cores, either the sequencing core, the metabolomics core, or the model organism core, to try to help them, obviously, in identifying the cause of this disease. Obviously, almost all patients and parents get sequenced, typically trios or quattros, and that information is then analyzed, and some candidate genes rise to the top and are then submitted to the Model Organisms Screening Center. We developed this program called Marvel that Shinya will expand upon, that is essentially mining the bioinformatic data that is already available in the model organisms and in the human genetic databases. This allows us then to prioritize the genes and assign them to a specific species, either C. elegans, Drosophila, or zebrafish. And as I said, I will mostly talk about what we do in Drosophila. So the success of the Model Organism Screening Center with respect to the Drosophila section is very dependent on the fact that we've generated numerous reagents that allow us to do elegant experiments and assess the function of human genes in flies and their variants. And so this all started by creating many 10,000s of mimic insertions in the fly genome. And when a mimic insertion is inserted in an intron, it allows us to introduce an artificial exon that consists of a splice acceptor, EGFP, and splice donor, and therefore will tag the protein, but the protein is mostly functional. 70 to 80% of the time, this insertion will make for a functional protein where that protein is tagged with GFP. We can also replace the mimic insertion with another exon that was developed by Ben White and his team, where you integrate a splice acceptor T2A Gal4 splice donor. And so what this does is will truncate the transcript, but during the translation, the Gal4 will be produced and the protein, the aminoterminal portion of the protein will be truncated. That Gal4 protein can then be used to drive the fluorescent protein and allow us to determine where the gene is expressed, drive the fly cDNA and show that it can rescue the mutation that we've induced, and finally drive a human fly reference or variant and assess if the variant behaves different than the reference. And so by doing this systematically for many genes in the genome, we've generated thousands of reagents and thousands of human cDNAs that allow us to do this systematically. And this has really been at the core of the MOSC. Now, in the past few years, this has allowed us to identify numerous new genes, and we've not only used this humanization strategy that I just described, that is kill the fly gene and replace it with the human cDNA. We've also sometimes used overexpression experiments of reference and variant and see if they are different. We've sometimes focused more on the loss of function studies and compare the phenotypes of the loss of function with the loss of function phenotype that is observed in the patients. But as you can see, it allowed us to identify quite a few genes so far. And as mentioned before, the MOSC has also been successful at identifying new human genes and participate in the diagnosis. So now I will focus on one gene, ACOX1, which is a case study that was just published in Neuron a few weeks ago or months ago. And ACOX1 is a protein that's present in peroxisomes and that affects the very long chain fatty acid metabolism and causes the demise of glia. And this work was done by Hyung-Lok in my lab. So ACOX1 is a critical enzyme in peroxisomes that is involved in shortening the very long chain fatty acids. So long chain fatty acids are turned into very long chain fatty acids. through an enzyme LOVL1, and then these are then imported in the peroxisome. And the first reaction is a shortening of this long chain to a shorter chain, and this leads to the production of high levels of hydrogen peroxide. And those high levels of hydrogen peroxide are diminished through catalase. And so this enzyme is a rate limiting step. And I'll first refer to you about the loss of ACOX1, which causes a very severe disease called ACOX1 deficiency. And I'll show you that the patients from the UDN do not have a loss of function phenotype, but a gain of function phenotype. So the patient that was submitted to the UDN had a single amino acid change, an N237S. Was a boy that had gradually at the age of 12 started developing an ataxia and recently died a few months ago. He did not have an increase in very long chain fatty acids, and you would surmise that if you lose the enzyme that shortens very long chain fatty acid, you should have an elevation of very long chain fatty acid, as is observed in the deficiency. And so a very different progression and a very different disease profile, but wheelchair bound with massive loss of Schwann cells. The loss of function is very different. The patients are diagnosed very close to birth. They die within a few years and have very elevated levels of very long chain fatty acids, an inflammatory response, and white matter demyelination. Now, we discovered a second patient through gene matcher in Korea and a third one in the United States in Cincinnati children. And so this provided us confidence that there was something important about this amino acid and de novo mutation. We first modeled the loss of function of ACOX1 using the strategy that I explained, where we introduce a GAL4. This then will disrupt the gene. This GAL4 can then be used also to drive a GFP. And the first thing we noted, which kind of was perplexing to us, was that ACOX1 was not ubiquitously expressed. It was actually only expressed in very few cells in the brain, and those were glial cells. So we also found that it was expressed in other cells like the immune system and the fat body, but in the nervous system, it was not expressed, mostly in neurons. So we looked at the loss of function of this enzyme in the wing margin, where we have these axons from the bristles of the wing margin that are surrounded by these glial cells. And when you look in the mutant, you see that, hey, there is a dramatic loss of axons and there is a dramatic loss of glial cells. And this is exactly the phenotype that was observed in this boy, that he had lost in the periphery his glial cells. Here we call them wrapping glia in Drosophila. In human, they're Schwann cells. And he had gradually lost these and therefore became ataxic. And this then led secondarily to an axonal loss. Interestingly, Locke then showed that bezafibrate, which is a protein that lowers the synthesis of the very long chain fatty acid by inhibiting this enzyme, ELOLV1, very strongly suppressed the phenotype. And so this was then suggesting that this might be used in patients for a cure.
3 more minutes.
So if we compare the loss of function in flies and humans, you see that the ACOX1 deficiency in human and the fly ACOX1 loss share many features. First, when we lose fly ACOX1, we have an early death. There's an increase in very long chain fatty acids. I didn't show you any of these data, but that is also true. There is a loss of glia. This leads to a loss of axons. That's also observed in both. There's a loss of vision in both. And finally, there is a strong autoimmune response in the human ACOX1 deficiency, and there is a very strong autoimmune response in fly ACOX1 loss. And so the loss of the syndrome in flies, the loss of the ACOX1 in flies nicely matches with what we observe in human. And the next question is obviously, what is the point mutation that is observed in the patient, and what is associated with that point mutation? So when Locke expressed this mutation in the fruit fly, the flies are essentially unable to move. They eclose, and that's about all they can do. They cannot eat, fly, move, and die. And as I mentioned before, the production, this enzyme, when it shortens very long chain fatty acids, leads to an elevated production of reactive oxygen species in the peroxisome. And this drug, N-acetylcysteine amide, blocks the production of ROS or scavenges ROS and leads to a beautiful suppression of the phenotype. Normally the flies are dead at day one, when there is no drug, when there is no N-acetylcysteine amide. But in the presence of the drug in a dose-dependent manner, you start seeing many flies that are surviving. This is also true in Schwann cells. If you express this,
1 minute.
This mutant protein in Schwann cells, you kill off most of the Schwann cells, as is shown here. And again, the drug nicely suppresses this phenotype in vertebrate Schwann cells. So in essence, if we come back and look at the gain of function and the loss of function, there's a clear difference, but in both cases, in human and in flies, they're very similar phenotypes. Severe reduced motility, there's no increase in very long chain fatty acids, there's no loss of glia, there's loss of glia, loss of axons, no autoimmune response, and antioxidants suppress the phenotype in flies as well as in humans. So this is essentially the last slide and shows the pathway. In ACOX-1 deficiency, bezafibrate is the drug of choice to suppress the production of the very long chain fatty acids. In the mutant variant, there is elevated ROS, and antioxidants are the drug of choice. So I'll end here and give credit to the people who've done the work. The MOSC, there's numerous people in our lab, in Shinya's lab, my lab, and Michael's lab that are participating in this. ACOX-1 story was done by Young-Ok Chung, and there are numerous physicians in the UDN that have helped us and provided patients. Thank you.
Thank you, Hugo. We don't have time to answer questions orally, but we can do that by typing it in the Q&A session. So Ada, can you try unmuting yourself and see if your audio is better now? Ada? Can you hear me? If not,
can you hear me?
Yes, we can hear you.
Now you hear me. Okay.
Yes.
Oh, it's weird. You might be on the phone. That's so strange. Okay.
Okay. So why don't we ask you to kind of restart from the beginning?
Okay. I will restart from the beginning. Let me find my talk.
Although we might not have time for the quick Q&A because we're kind of pushed behind. So
no problem. I'm going to start from the very beginning. Okay. So I am talking to you about the essential role of model organisms for clinicians. And you just heard an absolutely gorgeous example of why we need to work together all the time. And what I had started to tell you is what we do when we see a patient. So we see and evaluate a patient, and we try to determine if it's a recognizable syndrome. Once we do that by looking at OMIM, which I'm going to show you in a minute, then we send a test, or whatever test may be indicated, and then you may get a result that's the answer. You may get the result that I don't see anything that explains this, or you may get either a novel gene or a novel variant whose role is not clear in that patient's presentation. So we start with OMIM, and I have to tell you that I'm biased. I am the director for the last 18 years, but it is an online catalog of human genes and genetic disorders. Each entry has a stable number, and it has lots of aliases and full text descriptions that are structured. It's cited to the peer-reviewed biomedical literature. This is the structure of the data within OMIM. We have phenotype entries and gene entries, and they're linked through a gene map, and related phenotypes are entered into a phenotypic series. We have about 5,400 entries today, of which 16,300 are genes, and the rest are all phenotypes. Most are Mendelian, but we also have phenotypes where there's a major genetic contribution, for example, type 2 diabetes. We also catalog the growth of gene-phenotype relationships. This is data through the end of 2019, and you can see that it's continuing to grow, and it's growing rapidly. And some of the results are not nearly as clean as the beautiful data that Hugo just showed you. In a gene entry, we have standard information, and you can view the clinical synopses of the phenotypes associated with that gene as a table, which is what that shows you, and go through them. You can also see that we have particular variants In OMIM, the variants are example variants. We are not the variant database, but you can go to ClinVar to see a more comprehensive list of all the variants associated with this gene. And finally, there's something called the Phene gene graphics, which is a visual representation of the relationships between genes and phenotypes in OMIM. This you can toggle up, down. This is a PDF that shows what all these things mean, which obviously you can get from OMIM. And this is just what it looks like clean. And it shows you in a linear view or in a radial view. And this is the radial view will obviously rotate. The linear view open and close things. It's not, the linear is non-hierarchical. This is one of the phenotypes associated with ATR, which is Seckel syndrome. This is a phenotype entry. There are obviously different elements in a phenotype entry. This is an example. I went from the clinical synopsis, and you can go through it. And every place there's a blue bust. If you click on that, you'll see a picture of that feature. So if you don't know what micrognathia is, that is what this woman has, and that's a picture of micrognathia, not of the whole disorder, but of micrognathia. And those come from something called the Elements of Morphology. There are feature IDs for everything within OMIM, where it links to other resources such as SNOMED, UMLS, and HPO. And everything that's in OMIM is available through our robust API. And this is just an example of another side-by-side clinical synopsis. We have copious links, and they're topical. So there won't be a link from an entry to another resource unless there's a page about that about that topic in that other resource. So genome, DNA, protein, gene information, variant information, clinical resources for clinicians, animal model resources, and if we don't have something that you think we should, let us know. And then cell lines and cell pathways. We also have something called MimMatch. There's a 2.5-minute tutorial to tell you what that's about, which I'm not going to spend your time on right now, but the point is if there's a particular gene or particular phenotype that you're following and you want to know if there's something new, or if you do unusual searches within OMIM, you can do it and keep them in MimMatch. If we can't find anything in OMIM that helps us, then we go to GeneMatcher at genematcher.org, and it's a very simple resource to use. As of April 1, 2020, there are over 12,000 genes from over 41,000 submissions, over 9,000 submitters in 89 countries. About 57% of the genes have at least one match, and GeneMatcher's results in over 304 publications, and about 80% of them are novel disease genes. The graph on the lower part of the screen shows you the growth in the number of genes and gene matches over time. The divot that goes down in April of 2019 is when we culled emails that don't return, they're sort of dead emails. And on the right, you can see that there's already model organism researchers who are using GeneMatcher. Quite a variety. It's very funny that humans are listed as well, we are an organism. It's under a model organism subhead. But anyway, all kinds, remarkable kinds. So people are already using GeneMatcher. You can, within GeneMatcher, put in model organism matches. They will not, I do not think they go across to something called the Matchmaker Exchange. So the Matchmaker Exchange, GeneMatcher is a founding member of it. It's a consortium of different resources, databases that have largely human-based and case-based information from different research projects around the world. For example, the UDN feeds into Phenome Central, as does the Canadian Care for Rare. Decipher collects everything from their over 250 studies, but the largest one being the Deciphering Developmental Disabilities, Irish Japan, et cetera. So this is already a very large format for exchange of information. And it is linked to Monarch, which gets into information about model organisms. But one of the things that I think the world needs is a way to connect model organism researchers with knowledge of a particular gene or particular pathway with clinicians who might have patients with variants in those genes. Clinicians need model organism researchers when there are only a few patients with variants in the same gene, particularly those when those are missense variants or when the phenotype is highly variable among the patients. When there are missense variants whose effects are unclear, when we're looking for modifiers or to prove the effect of a variant on an organismal level, and anytime we're looking for therapeutic interventions, because a cell-based assay is very nice, but you really need to see what's going on beyond there. Finally, you're going to hear today, and you already have heard about several efforts in different regions to connect clinicians with model organism researchers. We need a system like the RDMM in the US We also need an easy mechanism to globalize the information, because outside of the coronavirus era, we were a very small world. Now we're very local. Linking clinicians with model organism researchers to validate observations and understand biology to develop therapeutics is essential and exciting, and we're very eager to work with you. Thank you.
Okay, thank you so much, Ada. We wouldn't have any time for answering questions orally, but if you have any question for Ada, please use the Q&A features, and she'll basically be able to type these answers for you. So thank you. The next talk is by Phil Hieter, and Ada just mentioned RDMM, and I think Phil would be able to tell you what RDMM is in much more detail in his talk.
Okay, I am going to try to share my screen now.
Yes.
Somebody else.
You should be able to share your screen.
Is it sharing?
No, not yet. Try again.
Okay. Share screen. Is that it?
Yes. Great.
Is that good?
Yes.
Okay. Well, thank you. What I want to do today is tell you about a national network of clinician and basic scientists that we've established here in Canada called Rare Diseases Models and Mechanisms, or RDMM. And the main goal of this network is to catalyze connections and collaboration between clinician scientists at the time they identify a new rare disease gene variant with a basic scientist who is poised to study the function of the gene in a model system. And this network was launched in December of 2014. So I think, as we all are aware, what the history books are going to show is that we're from sort of 1985 till, let's say, 2030, there's been this international effort to construct the genetic anatomy of human disease. That is, probably within the next 10 years or so, the scientific community will have essentially identified all of the genetic variants that cause human disease, or nearly all. And of course, we're sort of now in the middle of addressing the grand challenge, and that is, how can we understand how these disease variants cause disease? And that really comes down to understanding the function of genes, pathways, and networks that these genes are involved in. And through that knowledge of gene function, we really can have rational approaches to the prevention and treatment of human disease. So the target of discovery in human disease gene research is predicted to include something like 5,500 to 6,000 genes that will be involved in perhaps greater than 10,000 rare diseases. And currently, it's known, the disease gene variants for 5,400 diseases are currently known. These map to 3,800 human genes. And the guess is that there's going to be approximately another 5,000 disease genes variants discovered over the next, say, decade that might include another 2,700 genes, including the 3,800 that have already been found. So the rate of discovery now is on the order of a new disease gene discovery per day, almost on a daily basis. And I just want to quickly go through a little bit. Canada has been a significant contributor in the field, has been a leader in the field. And these have been already mentioned, that the efforts called Care for Rare and Forge, over a period of about 7 or 8 years, were involved in trying to elucidate the variants for about 1,000 rare diseases. And in fact, the Canadian clinician scientist community was able to be successful in about 50% of the cases. So approximately 500 genes or so were discovered over this period. And this has led to publications and many contributing authors. This has been really an outstanding example of a national effort of cooperation. And of course, when these genes are mapped to the cell, they map into many different functions. In fact, many of the complex phenotypes in humans are explained by intracellular genes that are involved in intracellular processes. So the challenge is really understanding the functions of these genes and how the variants contribute to disease. And this is just a figure that points out the fact that we really don't know a lot about many of the genes in humans. It shows, this is a plot of the number of publications per gene. And of course, there are few genes like P53 in which there are 9 to 10,000 publications, but there are 4,000 genes for which there are no publications. And about half the papers in the literature only account for about 5% of the genes. And this just emphasizes the importance of model organisms like yeast, worms, flies, zebrafish, and mouse, and other organisms, Tetrahymena, E. coli, and Xenopus, and other organisms that are required really to understand the function of human disease genes. So by understanding the function of the orthologs, the genes, the pathways, and networks, and through the fact that genes are so evolutionarily conserved, we can infer mechanisms of human disease and normal biology. And that's where the keys are to rational approaches to therapy and prevention. And so there's obviously a very active and vibrant research community that is involved in identifying genes, but you require also functional analysis. And of course, this is going to require increased collaboration, communication, exchange between medical scientists discovering genes and basic science who are involved in studying the function of genes. And that was the rationale for creating the national network, the Canadian National Network called RDMM. The basic objectives are to enable collaboration between clinician scientists that discover genes with basic scientists, as I said, scientists that are poised, that have expertise in the cognate gene's function. And what we wanted to do is to provide seed funding, a small grant of $25,000 to the collaborating scientists to obtain some initial results, either for publication in an initial paper, or as preliminary results for larger grants and things, longer term collaborations. So this is the network route map that you would find if you visited our website. And basically it shows that what we've done is we have recruited the model organism investigators in Canada to register their genes in a database, in a registry, and clinicians who are discovering genes can submit a connection application that goes to a clinical advisory committee that vets the causality, whether or not there's sufficient evidence of causality for the variant and the human disease. And then for those that pass these criteria, they're sent to a scientific advisory committee that have representatives of the various model organisms across the country. And they then can go into the registry and look for potential model organism researchers that have declared expertise in the orthologous genes. And if these are found, the scientific advisory committee can invite the model organism researcher to submit a grant. And then within a short time frame, we review these grants. And the hope is that by doing this, we can validate gene discoveries. We can generate data for, oops, now I've lost this. Let me go back. For publications, we might give insight to disease pathogenesis, suggest potential therapeutics, et cetera, and foster longer-term collaborations. So the core activity of this network is to provide these rapid response grants and seed funding. And so the way this works is the clinical advisory committee receives requests twice monthly. These are vetted within a week. The SAC receives from the CAC any genes, and we search the registry, invite labs to apply. There's a 2-page application and a brief CV, and the SAC reviews us within a week. So the entire process takes about 4 to 6 weeks. And if we successfully make a connection between a clinician and a model organism researchers, we provide a $25,000 seed grant. Challenges in setting this up was creating a national database of model organism researchers and the genes that they study, and recruiting participation for the entire community. And through the work of our bioinformatics team, Paul Pavlidis and Sonia Rajek, they created a really nice registry where model organism researchers could create an account, add basic information, the model organism that they work in, and add their genes and Go terms, which is a way of identifying them. The genes are added in 3 tiers. So tier 1 are genes that are actively being worked in the lab. Tier 2 would be other genes that are related to the tier 1 genes, maybe proteins that interact or within the same pathway that the lab could easily functionalize in their labs. And then there's a tier 3 category in which the database suggests Go terms related to the genes in tier 1 and 2 that the researcher has submitted, and provides you know, a 3rd tier within the Go term. So just to give you an idea of the flow over the first 5 years of this effort, there were 135 genes that came into the clinical advisory committee. 95 were passed along to the bioinformatics core, and in addition, we added 116 other genes that had been identified previously in the Care for Rare, and actually some from the Model Organisms Screening Center at Baylor. So for a total of 211 genes that came through the bioinformatics core. 140 of those genes had a potential match in a model organism researcher lab in Canada. These went to the Scientific Advisory Committee. Of those, 101 genes had investigators that were invited to submit a short proposal. Of those proposals, 77 of the genes had at least one grant or Catalyst Award funded. And so that these 77 genes actually had 85 model organism Catalyst Awards. So I think 80 of the genes actually had 2 independent awards in 2 different model organisms. So this is basically how it goes. In addition, we've considered additional 46 proposals in targeted calls in partnering with patient advocacy organizations, and we funded and co-funded some of those. So I won't go through, I'm not going to go through, there's obviously many examples, but this just shows 2 where there was a severe development delay in skeletal dysplasia disorder. And there were, in this case, 2 independent grants were funded, one in Drosophila, one in zebrafish. And this led to a really high profile paper in Nature Genetics. And then a second example where there was a link to a worm lab. And again, this led to a very nice paper. And I won't go through the biology, really focusing on the network itself. So just to give you the statistics to date, there are 574 researchers, model organism researchers that are registered in our network. That represents, we think, about 85 to 90% of the PIs in Canada. The number of genes that are added, there are 7,250 genes in tier 1 and 2. And if you include tier 3, there's 12,377 unique genes in model systems.
3 minutes.
That would require, that essentially corresponds to orthologs of about 7,725 human genes. So over the 5-year period, we've been able to make 91 connections between clinicians and model organism researchers. So again, I pointed out that these were the type of outcomes we were hoping for: validation of this gene function, high impact publications, potential rationales for treatment, and increased success in obtaining grant funding. And we have strong examples in each of these outcomes. An additional indirect effect that I think is important is that it has generated increased awareness of the importance of basic research in model systems to funding agencies, the community, patients, clinicians, and the general public. So what about in the future? And we've had a renewal of our grant now for the next 5 years, starting in 2018. And we've moved to try to coordinate and collaborate with other efforts internationally. So we've shared our infrastructure with groups in Japan, Australia, and Europe. And we're hoping to try to establish a network of networks. And the idea is, as genes come through our system, I think as I said, there were about 2/3 of the genes failed to get a match. So it would be possible that these genes could then be used to search in the registries of other RDMMs in other countries, for example, in Europe, Australia, or Japan. And these are now ongoing efforts that are being established in these countries. And the idea is that if we can, over the next while, hopefully soon, these independent RDMMs can be communicating with each other and be sharing, doing cross registry searches to try to make connections across borders. And Europe is now online. They've begun an RDMM effort. Japan has an effort. They've initially started really working within the system, but we're working with them to try to expand that. And
1 minute
is close to opening there. So I'll end there. And just these are acknowledgements of the people that have been involved. So thank you.
Okay, great, Phil. Thank you so much.
Yeah.
Again, we're kind of pushing late, so there was no time that we can answer oral questions orally, but people are having questions listed on the Q&A session. So if you can look at those and type in your answer, that'll be great. Okay. So the next talk is going to be another person from Canada, Lauryl Nutter, who's going to be telling us about the IMPC, International Mouse Phenotyping Consortium, and how that could facilitate collaborative research. Lauryl?
Thanks, Shinya. Whoops, let me just start the right program here. All right. So unless I hear otherwise, I'll assume you can see my slides. So thank you, Shinya, for inviting me to give this talk. And I hope this can provide some of you with some information. And also, I hope spark you to access the resources of the IMPC in either your basic research or your clinical research. So what is the IMPC? The IMPC is the International Mouse Phenotyping Consortium, and it's a program that's set up to generate null mice for high throughput clinical screening. We partner with external stakeholders to prioritize genes and also to add value to the resources we create. And really we're set up to explore the dark genome. For those of you who have tried to write grants about genes for which there is very little information known, you know it's really hard to get that initial grant or to get that initial funding to sort of prove your first hypothesis that this gene is important. And as the previous graphic showed, only 5% of the genes in the genome account for 45% of the publication. So expanding our view of the genome is really important, and the IMPC really is trying to help do that. And so how are we doing that? So you all know that human and mouse share a lot of homology in their protein coding genes, in their orthologous protein coding genes. So the first effort started in the early 2000s with the International Knockout Mouse Consortium, whose goal was to produce a publicly accessible library of targeted mouse ES cells. So their goal was to make a knockout for every protein coding gene in the mouse genome. After the IKMC ended, that was a 5-year effort, the International Mouse Phenotyping Consortium started, and we were undertaking the conversion of those ES cells into mouse lines and then the phenotypic analysis of those mouse lines. So what happens to a whole organism when you remove the function of this gene? More recently, we switched to using Cas9 to derive our mouse lines because it's much faster and more cost effective. And it also enables us to respond more nimbly or more agilely to requests or expressions of interest in genes. And so to date, we've provided phenotype being information for 6,440 genes. And what have we produced? So the phenotyping obviously lags behind the production. So we've produced to date over 8,844 model or knockout mouse lines in unique genes. So there's about 9,500 mouse lines in total, but that accounts for 8,844 unique human ortholog genes that we produced. About 5,100 have been made by ES cells and about 3,700 using Cas9 technology. I'm not going to go into the details of what these alleles look like because that's available at the IMPC website and elsewhere. So what do we do once we have these mouse lines? Well, we put them through an extensive broad-based phenotyping. So from the age of 4 weeks to 16 weeks, we're looking at these mice initially just with a weight curve, but then we work on doing tests to assay several areas of function. So including neurobehavior, musculoskeletal, learning and memory, metabolism, cardiovascular, sensory systems, the immune system, and then finally doing full pathology at the end. So how can you use this data? So IMPC has both formal and informal partnerships with several consortia, including the Centers for Mendelian Genomics, the Undiagnosed Diseases Network, Care for Rare, the Province of Ontario Neurodevelopmental Disorders Network, and also the Gabriella Miller's Kids First program at the NIH. And we also have partnerships with individual clinicians and scientists. So how do these partnerships work? Well, they're fairly flexible and they vary depending on whether it's a consortium or a scientist and how many genes are of interest to that group or individual. And it really starts by nominating genes. So there's a place on the IMPC portal where you can nominate or express interest in a gene or follow a gene. Once that gene is nominated, it gets prioritized and assigned, and a production center can pick it up and produce the null mouse line. Once that null mouse line is produced, it is subjected to broad phenotyping, and as shown on the previous graphic, we phenotype 7 males and 7 females. Homozygotes if they're viable, heterozygotes if they're not viable, and if they're not viable, then we do some embryo phenotyping, and I won't go into those details, but it essentially involves 3-dimensional imaging of embryos. And once we've completed the broad phenotyping, we disseminate these resources. So the IMPC website, we post all of the data as analyzed data, but you can certainly go and download all the raw data if you're interested in it. And we also make the mouse lines publicly available through a variety of public repositories for mouse lines. So sometimes, and sometimes this broad phenotyping through a matching program that was developed by Damien Smedley and his colleagues in Monarch, actually the broad phenotyping can give us a validated disease model. However, sometimes if there's additional funding, so that's the core IMPC funding. With other funding, sometimes we make specialized alleles instead of knockout alleles, and this is something that we're doing with the Kids First organization right now, for example. And then those alleles can be subjected to our broad phenotyping, or many of the centers are also able to do specialized phenotyping. And this data can get added into the mix to produce a validated disease model for a given human disease. And often these specialized alleles are point mutations, missense mutations, or nonsense mutations. Occasionally they are deletions of enhancers or other non-coding regions, so they can vary quite a bit. And so I just wanted to give you an example of one a couple of our partnerships. So this one is really around the disease autism spectrum disorder. So this is not a rare disease, this is a common disease. And this is with the Province of Ontario Neurodevelopmental Disease Network. So they provided us with gene candidates that they identified by GWAS, the literature, or through disease associations. Then we produced the exon deletions at our high throughput production center. And then we did heterozygous by heterozygous crosses. And if they were viable, we analyzed the homozygotes, as I mentioned, through our clinical screen. And if they were lethal, we analyzed heterozygotes. At the same time, with funding from POND, we produced a second cohort of animals that went through their neuroanatomical screen, which is essentially whole brain MRI of 8-week-old mouse brains. And so I want to give you an example of one of the outcomes of this. It was a gene called NHS, which is linked to Nance-Horan syndrome. So these null mice are viable as homozygotes. So we studied homozygotes. In Nance-Horan syndrome, it's the characteristics are intellectual disability as well as facial, dental, and vision abnormalities. But this gene has also been linked to autistic traits. So in our phenotyping pipeline, what we identified is abnormal locomotor behavior. So the mice, the homozygotes were less active, as shown by these whisker plots in open field. And we also saw abnormal retinal morphology. So both of these correlate with some of the phenotypes identified in the human patients in NHS syndrome. But what was interesting on the neuroanatomical screen is they saw an overall brain volume increase of 10%, which is the largest volume increase of brains they've ever seen other than for P10. So it was a huge difference. But what's interesting, even though the brain was larger overall, it actually had a decrease in cortical structure. So on this heat map, the reddish pink is shows where structures that are larger, and the blue shows structures that are smaller. So these cortical structures are smaller, which could be linked with intellectual disability. So they gave us actually about 25 genes to look at. And so some of the first genes that we managed to get through is nearly all of them actually ended up, they showed behavior differences in the mouse for most of them, except for RALY, which only had a cardiac phenotype in the mouse, identified in our pipeline. And they saw brain volume changes in almost all of them, again, except for RALY, but they did identify a structural cardiac phenotype. So that's actually gone off to different people who are interested in studying that gene in respect to cardiovascular development. So this has been really successful for them, and they have several papers that are in the works and should soon be published, including one on ARID1B and one on another gene that's not shown here at this time. So that is one example. Another example is Care for Rare, also nominated several genes to our pipeline. And in this one, LRRC56, they'd identified this in a syndrome in a disease that could seem to cause lethal prenatal situs inversus in humans, as well as there were some patients who survived but had recurrent respiratory infections. So we made the knockout mouse, and we put it through our broad-based phenotyping, one of which is ECG. And for those of you familiar with ECG traces, what you'll see here is that it looks like this ECG trace in the homozygotes is actually upside down. Now, remember, these mice are running around and looking perfectly happy and healthy, but their ECG is upside down. So we did an echocardiogram, and what we saw is that the heart is actually reversed. So in a wild type mouse, here's the right versus left ventricle, but in a homozygous LRRC56 null, it's reversed. The right ventricle is on the left side and the left ventricle is on the right side. And so what we think is that this inverted ECG is likely a result of the heart being reversed. And so when we open these mice up at pathology, we see a partial situs inversus. So the heart is often reversed. The kidneys, usually one kidney is higher than the other. I think it's the left is usually higher than the right. I could be wrong. That's reversed. The liver seems to be partially reversed. The lungs seem to be reversed in some of these animals. So it's a variable phenotype. But the nice thing about this in terms of the situs inversus, but the great thing is that these mice survive. So we actually now have a model that will enable some mechanistic studies as well as examination of the infection phenotype. So they had previously identified that this protein was localized to the flagellar tips in a unicellular organism, but now they have a mammalian organism in which they can study some of the things that are going on with this disease. And so I'm going to stop there and just acknowledge the people who do most of this work, which is for the mice that I've presented, the TCP model production and phenotyping cores. Jacob Elgood and Jason Lurch at the Mouse Imaging Center at TCP did the neuroanatomical screen. And then of course, all of my IMPC colleagues, particularly the other members of COMP who are involved in model production. And of course, the funders who help us do this work. And with that, I'm happy to take any questions if there's time, but if not, I can answer them otherwise.
Thank you, Laurel. Yes, you were perfectly on time, but because the whole session is kind of pushed back, we'll actually move on to Andy Golden stock. And if you can look at the Q&A session, people would actually be typing in questions through there.
Okay, great.
Thanks. So next, yeah, thank you. So the next two talk would be actually C. elegans. So Andy would be telling us about modeling rare monogenic human diseases in C. elegans. Can you share your screen?
I'm trying to do that.
Yep. There's a green button.
The whole thing just went blank.
Yeah. Oh, there you go.
Can you see me?
Yeah, I see your desktop. Yeah.
Okay.
Oh, well, I actually got canceled. Maybe do it again.
Okay, let me just get back to the beginning.
Can Can you try
Okay.
sharing your screen again?
Can you see it?
No, we can't. So
Okay, I can start.
no, well, we can't see your screen.
Crap.
Oh.
If you hover over the bottom, there should be a share screen with a green button with the upwards arrow. Do you see that?
Yeah, but when I click on my screen,
Yes, now we see you.
oh, sorry.
Great, thank you.
Okay. I want to thank GCA for pulling this off, and also Shinya for organizing this session. So I'm going to talk about modeling rare and neglected monogenic human diseases in C. elegans, and I just want to give you 3 quick examples of what we've been able to do. And I think almost any lab with any gene of interest could really tackle such problems. And so our criteria to really model any rare monogenic human disease in the worm is just to simply find the nematode ortholog, which we've had a great deal of success in. I would say about 60% of the disease-causing genes we could find in the worm. We then, as a preliminary start, delete the entire gene using CRISPR-Cas9 and just characterize the null phenotype, just to tell us that there is something to follow. We then go along and make patient-specific missense alleles, for which in every case that we've looked at, we can find at least a few amino acids that are absolutely conserved, and therefore making patient-specific alleles isn't difficult. And then we characterize those patient-specific alleles. And I have this highlighted because that's often the biggest challenge, is that we don't, for obvious phenotypes, yeah, not a big deal, but for some things, it's subtle, and then we might have to run many numerous assays to figure out what's actually wrong with these animals. And then from there, we can do functional study of genes at both the cellular and molecular level. My long-term goal is to actually do suppressor genetics, where we do suppressor screens or chemical suppressor screens to hopefully get at therapeutic strategies. Our gene discovery mechanism is really that we either find a gene that we learned about in the literature or that a colleague suggested. The other advantage at the NIH is we actually have access to the Undiagnosed Disease Program candidate list that gets sent to us every month. And from that list, we can pick and choose genes that look interesting to us. And we also have the benefit of attending clinical rounds from the Undiagnosed Disease Program every week. And in these cases, we actually go to a lot of case reports where N equals 1, and we actually get to meet the patients and learn more about that disease in person. So these are some of the syndromes that my lab has started to work on. There's a full range of cardiac arrhythmias to autoimmune diseases to lipodystrophies, and down here in the bottom 3, we've even worked on some craniofacial diseases. And so I think that for many of these, you can see it wasn't hard to find the worm ortholog. So I just want to give 3 quick examples of some of the things we've been able to do. CPIN is a gene that's involved in lipid droplet biogenesis. It causes lipodystrophy in humans. We could easily knock out the entire gene using CRISPR. We did this in this allele, as well as 2 other alleles. And what we found is that where wild-type worms have almost 100% embryonic viability, these 3 deletion alleles were somewhat subviable. Now, given that this was involved in lipid droplet biogenesis, we thought that these animals might be osmotically sensitive because there's a lipid lipid-rich layer that's important for permeability. And so if you take a wild-type embryo and stain it with DAPI, you only can permeabilize enough to get the polar body stained. However, if you do our mutant embryos, you can see that all the internal nuclei stain quite well with DAPI. This is also true with a number of dyes. These embryos are permeable, where wild-type embryos are not. This is an M-cherry fusion to an outer eggshell protein. And what you can see here, this black mark here indicates the permeability barrier. And this separates the outer eggshell from the embryo proper. And as you can see in our mutants, that permeability barrier is now no longer existent. When we go to EM, you could see this permeability barrier quite clearly shown here, separating the outer 3 layers of the eggshell from the embryo proper. In our mutants, you can see the outer 3 layers, with one of them being quite expanded, and now they touch the embryo proper, and there's no permeability barrier. So we believe this explains the reason for the embryonic lethality. These embryos are completely permeable. We tried a cute little study where we tried to see if we can rescue the permeability by supplementing the diet with gamma-linolenic acid, a fatty acid. And as you can see here, where this has no effect on wild-type embryos, on our mutants, it makes them impermeable to dyes. And so these animals are somewhat rescued and have a much better eggshell. In another example in my lab, we studied a multiple mitochondria dysfunction syndrome, MMDS1. This is one of six such syndromes. All 6 of these syndromes have perinatal lethality, where these kids die at about 12 to 15 months. They're all hypotonic, and they have impaired respiration. All 6 genes are identified to date disrupt the iron-sulfur cluster delivery to recipient proteins. Iron-sulfur clusters are metallic cofactors that are found in a lot of the electron transport chain proteins, as well as some cytoplasmic and nuclear proteins. Collectively, this is a very rare collection of diseases where there's only about 100 cases reported. We studied LPD8, which is a C. elegans ortholog of NFU1, the gene that's mutated in MMDS1. This is the iron-sulfur cluster binding domain, and you can see it's conserved. And marked in yellow are all the patient alleles that are disrupted in the C terminus between yeast, worm, mouse, and human. And shown here is just a ribbon diagram that all these patient alleles are really intimately associated with the domain that binds these iron-sulfur clusters and thus delivers them to recipients. Peter Kropp in my lab made 5 patient alleles that are absolutely well conserved in C. elegans. And what we found is that these are all very slow developing animals, and 4 of the 5 alleles cause sterility, and we have no broods from 3 of them because they're completely sterile. And we also found that when we did lifespan assays, that the majority of these animals also have a reduced lifespan. Since this is a mitochondria disease, we wanted to look at mitochondria morphology. And if we look at the syncytial germline, you can see very punctate mitochondria surrounding germ cell nuclei. However, if you look at one of our severe mutants, the mitochondria are completely swollen and occupy all the surrounding region around the nuclei. We've also observed this in the hypodermis and in the muscle, and we believe this is ubiquitous mitochondria phenotype. We can also measure oxygen consumption, a proxy for electron transport complex function in real time in living animals. And so we can inhibit as well as stimulate different electron chain complexes to access the functionality. So in our most severe LPD8 alleles, 3 of them shown here, we have dramatically low basal respiration, shown here, and this is wild type. And when we try to stimulate maximum oxygen consumption, you can see that these alleles don't respond anywhere near what wild type does. And thus indicates that there's severe electron chain complex dysfunction. And in the third disease I just want to highlight, we're all start studying this autoimmune disease called COPA syndrome, which we learned about from attending the Undiagnosed Disease Program clinical rounds. We had a patient presented there, and that interested Isabella in my lab to pursue COPA. COPA is the alpha subunit of the codomer complex, which is required for retrograde Golgi to ER transport of dilysine tag proteins. This is an autoimmune lung, joint, and kidney disease. And what was interesting is that all of the patient alleles map to this 14 amino acid region. This region is absolutely conserved between humans and worms, as you can see. And Isabella has begun to make all of these analogous patient alleles in the worm gene. Interestingly, if we delete the COPA gene entirely, embryos die as homozygotes. If we delete just that 14 amino acid region, The embryo is also a dais homozygotes. But all of the point mutations that Isabella made so far are completely viable and look normal. And so this is a case where now we want to look, is there some subtle phenotype that we're just missing because these animals are viable and fertile and everything looks normal about them on the plate? And so Isabella decided to give them a little ER stress. And we found that these animals are hypersensitive to ER stress. So using a BIP ortholog called HSP4 fused to GFP, this is a good indicator of ER unfolded protein response. And in wild type animals untreated, you can see that there's a little GFP expression in our mutants as well as in wild type animals. However, where wild type is totally tolerant to this low dose of ER stress, where you can barely see any GFP, you can see that our mutants are hypersensitive to this ER stress and light up the BIP1 ortholog, suggesting that these animals are very sensitive to ER stress. And this is a mechanism to now uncover some other phenotypes. I don't have time to go into another example, but I do want to point you to poster 1954A. This is a mechanosensitive calcium channel called piezo that's implicated in, I think, 5 or 7 different human conditions. And what's really cool about this is that we made this mutant and it has a really dramatic ovulation defect where when oocytes are ovulated into the spermatheca, the spermatheca actually crushes the oocytes and gives us a very reduced brood size. So to conclude, I think I can convince you that worm is a great model organism. Deletions of the orthologs that we've worked on all have had phenotypes. We've made patient-specific missense mutations. They often have dramatic phenotypes though, like COPA, some of them we had to work a little harder at uncovering. We're now in a position to suppress these phenotypes doing genetic suppressor screens or chemical suppressor screens. And we hope that this allows us to learn more about the cellular phenotypes of each of these disease-causing genes. Just want to thank Peter Kropp, Pippa, and Kyle for the LPDA project, and that was done in collaboration with Michael Sachs' lab at NIH. And the COPA project was all by Isabella, and the CPIN project is Xiaofei Bei, also in collaboration with Chaowen Wang in Taiwan. And we have a number of other projects in the lab, as well as current and ex-students. And I do want to thank Bill Galt at UDP for including us, and the Undiagnosed Disease Network for also including us. If you have any questions, I have a very simple email address. Please feel free to email me if there's not time to address these questions. Thank you.
All right, thank you for the great talk, Andy. Again, we actually are getting close to catching back on time, but let's actually have all the questions and answers done on the Q&A form, so that if you can actually take a look at, there are already some questions for you addressed there.
Okay, thanks.
So if you can look at that and answer it, it would be great. So the next talk would be by Alex Parker. And he's from Canada. So model organism-based drug discovery pipeline for amyotrophic lateral sclerosis. So we're going to be moving on from diagnosis, mechanism to therapeutics here. Alex? Can you hear me, Alex?
Muted.
Oh, yeah.
Hold on.
Yeah.
Okay. You can hear
Please.
me?
Yeah, now I see you. Can you share your screen, please?
Yes, I will do that right now.
Yes, I see it now.
Perfect. So, okay. First of all, I'd like to thank Shinya and the GSA for even making this possible. I'm happy it's happening. So I'll tell you about a story that's been going on for several years using model organisms for drug discovery. And before I get to that, just in terms of full disclosure, I'm part of a company, a founder for using model organisms for drug discovery. And some of that came from our work, where this is a collaborative effort by several labs, researchers, basic researchers, and clinician researchers. This was all done in Canada and mainly in Montreal. And a number of us had decided at the time to make models for amyotrophic lateral sclerosis, or ALS, and we combined them together in terms of developing a pipeline for drug screening. And a lot of this data is published. I think almost all of it is, and you can go to this paper to get more information. So I'll go through the story, and then maybe let's give people an idea of like what's involved to start from worms and end up with clinical results. Hope you find it interesting. So as I mentioned, the disease in question is ALS. My lab is interested mainly in modeling late onset degenerative diseases, and ALS is one of them. It's a motor neuron disease, also called Lou Gehrig's disease. You know it from the Ice Bucket Challenge, of course. And it's a really aggressive disease. Many of the patients die within a few years of diagnosis. No treatment, no biomarker. Most of the time it's sporadic, but the genetics is advanced, and there's 10% of familial cases, and there's more than 30 genes, but you can group them into like the big 4 genes, I suppose. This would be SVD1, TARDBP, encodes TDP43, FUS, and C9orf72. Now, while the genetics is like been really advancing, the number of genes involved seems to be doubling every 4 to 5 years. This is great, but it's still a little complicated because with all these genes and proteins, it's not clear exactly what the overall function is in the context of disease pathogenesis. And this is a review from a few years ago. You can group these proteins into like protein homeostasis. RNA binding proteins, of course, and cytoskeletal proteins are involved. While that's interesting, in terms of drug discovery, it's still a little complicated because if you're looking at a traditional classic target-based drug discovery mechanism, you need a target, you do screening, you find molecules, you optimize mammals, clinical trials. But there's no consensus for what the main mechanism is involved in ALS, so you can't really decide on any particular target. So an alternative approach is a phenotype-based drug discovery, where here you take your favorite model organism, and you construct a model. Hopefully you have a phenotype, because if you don't have a phenotype, you're out of business with that particular model usually. And then you use this to screen for lead molecules. An important point here is that with all the tools, genetic tools and additional tools available to the model organisms, once you have a hit, you can use these to go forward and try and find what the mechanism is. Find a gene or a pathway that is required for drug activity. Use this for lead optimization, and then from there you go to mammalian model testing, and hopefully with this like extra validation in biological systems, it's more relevant to the clinical setting. That's one hypothesis anyway. So thanks to Andy for a great talk about why C. elegans. I don't need to convince anybody, hopefully, like why you would use model organisms or worms for that matter. But I'll just quickly describe the model that we used several years ago now to start this. And it was a classic transgenic approach where we took human cDNAs from that cause ALS in patients, and here encoding TDP43. Since it's a motor neuron disease, we expressed it in the motor neurons with UNC47 promoter. We've since gone on to make CRISPR versions of these type of things, but this is the model I'll be talking about today. And for use in this, we needed to have a phenotype. And luckily, we had some decent phenotypes. First of all, these worms, this can be the TDP43 or FUS. We see this for most of our ALS models for the most part. They develop normally, but as they get older, they don't move very well when you grow them on plates. And then it's a worm not having a great day. If you come back and this worm's aged a little bit longer, it doesn't move very well at all. So this is a very easy phenotype to see on plates. These animals become progressively paralyzed, and at about the same time, you can see that their motor neurons begin to degenerate. And you can see this, we score these basically as gaps or breaks along the processes. It's age dependent, and it occurs at about the same time as the onset of these motility problems. So that's great, we have a phenotype, but something that made it even easier for us, and this is perhaps the trick of the system, is that when you grow these worms in liquid culture, you get this paralysis developing acutely. Now, for those of you who don't know, you can usually grow worms in captivity, I suppose, in the lab or grown on plates, solid media. You can also grow them in liquid culture where they have this stereotypical swimming, thrashing motion. And these are wild type worms swimming in a 96-well plate, and these are the mutant ALS strains. And you can see they're not moving very well, and they're basically paralyzed like you would see them on plates. Now the bonus here is that this phenotype, which you usually see developing over 10 to 12 days when grown on plates, you can see it in a matter of hours. So this opens the door to same-day screening, which is what we did. We just went ahead, and this is several years ago now, we collected many of the commercially available drug libraries available at the time. We pulled them together, and we went ahead and did a phenotype-based screen based on motility. And here is what I always say is a movie of worms not moving very much. And you can see that they're barely moving. So this is the animals blocking out after several hours. And this is an example of a hit. And the first time we did this, we just used their eyes, but we've since gone on to automate and streamline the procedure. And this is something that we're doing in the context of the company as well. But the next part was very important. It's like we wanted to make the jump from worms, getting closer in these iterations to a clinical setting. So my colleague, Pierre Drapeau here at Université de Montréal, the CHUM, he had developed zebrafish models at the same time as we developed the worm models. And we thought perhaps we could take our lead compounds and test them in a vertebrate, in a fish model, to really identify the most active molecules. And this is a summary of the screen at that time. We took a number of chemical libraries, around 3,600 compounds. We screened them in the worms, primary, secondary screening, and we came up with a list of 13 molecules. And these are structurally and or functionally related neuroleptics or antipsychotics. And of this group, the ones in bold were then tested in another phenotypic screen, this time in zebrafish. And I have a movie of that in a second because I like it. And from here, additional screening, we ended up with a lead compound called pimozide. Now, pimozide is an antipsychotic drug. It's been used in schizophrenia. It's been around for a long time. It's generally known as a dopamine receptor D2 antagonist. It does have many off-target effects. But one mechanism that we thought might be of interest to us is that it may be acting to facilitate transmission across the neuromuscular junction. And this is what has emerged as a theme in all of our ALS models, is that perhaps what's going wrong here is that the function at the neuromuscular junction is disrupted, and maybe this is relevant to ALS. So when we get to what we call a lead compound, we decided it's a lead compound because it also works in additional assays, motility, neurodegeneration, aggregation. It also worked in additional zebrafish models. And finally, I'm not talking about this today, but we've also done screening for pimozide derivatives. Now, here's an example of validating pimozide in a zebrafish model. These are zebrafish embryos expressing wild type TDP or mutant. It's a touch-evoked escape response. And if you tap it, it's, okay, so it moves and then, oops. And the next one doesn't move very well and is partially rescued with pimozide. All right. And you can see this with the traces. Now, something that's interesting is that complementing the worm work is, I guess you could say zebrafish is a little more accessible for the electrophysiology. So Pierre's group had went on and measured neuromuscular junction activity by looking at end plate currents, and this is disrupted in the TDP43 fish and is rescued partially by pimozide. We then went on to recruit our local collaborator, Richard Robitaille, who had been working with an SOD1 mouse model of ALS using additional electrophysiology techniques. And he's measuring end plate potential and quantal content in these animals, and it's decreased in the mutant, and pimozide can rescue it as well. So this is great because this drug seems to be working at the neuromuscular junction in 3 animals, worms, fish, and mice. And from here, we're really lucky to come in contact with a clinician researcher, Lawrence Korngut at the University of Calgary. And even luckier is that he had funding from a private source to run a clinical trial. And I didn't mention this explicitly, but pimozide is approved. It's been approved for many years, and it's relatively cheap as a drug. So he decided to go ahead and run a clinical trial. And it was a relatively, I guess, small trial, but still, 25 patients, 6-week treatment. And this is the study timeline if you're interested. The patients were started at 6 milligrams and 10 milligrams, but this had to be dropped to lower concentrations during the study because it was the patients did show very strong side effects. And at the end of the study, a number of metrics were used, and there's a number of scales that people are using to measure this type of things. ALS functional scale, no effect on slow vital capacity. However, they did see improvement in something called the MRC sum score. And this is a number of assays that look at motility ranging from paralysis to normal strength. So pimozide managed to stabilize this, whereas the patients on the placebo got worse over the course of the study. Now, finally, the question of like neuromuscular junction transmission as being a target. So this is the clinical translation study. And so there's something called repetitive nerve stimulation, which is a way to basically fatigue the neuromuscular junction. So a decreased response here, that you see this happening in some ALS patients, may be an electrical correlate of clinical muscle fatigue and weakness. So perhaps if you see this in the patients, can you correct it with pimozide treatment? This would give a direct proof or a potential target engagement of this drug working in the way that we think it's working. And it could also be a biomarker of disease treatment or progression. So Lawrence looked at this and they focused on a muscle in the hand, this thumb muscle, and I learned a lot as we got closer to the clinical reality of disease. And what they noticed was patients on the placebo, this is one of these compound motor action potentials reading, patients on the placebo got worse by the time the study ended, whereas the patients on pimozide, either low dose or high dose, stabilized. So in this instance, the drug was stabilizing symptoms in patients, and we're very happy with this. And thumbs up for ALS. And this has now gone on to a second national clinical trial, 100 patients, 6-month treatment, and we should know the results in the next year or so. So finally, I'm almost done here, is that what we think the mechanism is that pimozide works by stabilizing neurotransmission right at the top, and blocks all these downstream effects. So I'm going to end on this slide because I think I'm over time, and to thank all the people who were involved in this from the beginning, many, many people. It's a long project, and we got it all together in one publication, so you can read it there if you like. So I'll stop there.
All right, thank you, Alex. Actually, you were perfectly on time. So.
Okay.
Yeah, but it's a great talk, and I think I really like the fact that you went from C. elegans not directly to humans, but you used all the intermediate zebrafish mouse to kind of really translate your findings all the way to the clinics, and hope the clinical trial would actually give you, or the patients actually, positive results. Right. So again, just like the other talks, why don't we actually do the question and answers on the chat, the Q&A tab, so that we are all consistent for this session. And actually, the last talk is by me.
Want me to introduce you?
Please, please.
This is Shinya Yamamoto from Baylor College of Medicine, and he'll be telling us about how to connect with clinicians.
Great. People see my screen?
Yes.
Okay, great. So thanks, Andy, for the introduction, and thank everybody actually again in this session for the wonderful talk. So after listening to all the great talks about how productive collaborative research can be, many of you probably start to think like this person here saying, I really want to start looking for clinicians who I can collaborate on my favorite gene, but where do I start? I think there's 3 important steps that you need to take. First, you need to gather information about your gene of interest in other species, including human. Second, you need to try to find clinicians who are interested in the human ortholog of the gene of interest. Third, you need to initiate communication with the clinician and establish a working plan and a trust relationship. Today, I would discuss these 3 points, including some useful internet resources that can help facilitate these processes. The first thing you need to do is to gather a lot of information about your gene of interest. Obviously, you have significant knowledge about this gene in the specific model organism you work with, but you also need to know additional things like, what is the orthologous gene in human? Is the human ortholog associated with any rare diseases? If not associated with any diseases yet, what is the likelihood that deleterious variants in this gene may cause novel disorders? You also want to know what is known about this gene in other model organisms. To do this effectively, you can take advantage of different database integration tools that combine many different kinds of information from multiple data sources. Tools including Marvel, Gene2Function, Monarch Initiative, and Alliance of Genome Resources are particularly useful for this purpose, and these websites are all complementary to one another. For example, Marvel is a tool that we have been developing in collaboration with Zandon Liu's and Hugo Bellen's lab at Baylor College of Medicine. Marvel allows users to quickly identify human ortholog candidates of model organism genes, collect information about human variants associated with rare diseases and those that are found in healthy people, and further provides information on gene function and expression in humans, as well as in 7 key model organisms. To use this website, you can visit the marvel.org website. Note that there are 2 R's here, and click the tab on the top that says Model Organism Search and enter the official symbol of your favorite model organism gene to initiate your search. Marvel would first give you a list of human ortholog candidate genes, which is provided using the DIOP program developed by Claire Hu and Norbert Perelman's lab. Here, I searched the Drosophila notch gene, which pulls up notch 1, 2, 3, and 4 in human. To further obtain information about each human gene and their orthologs, you can click on this Marvel It button that are shown on the right to proceed to the next step. Clicking on notch 1 would give you a 1-page summary of information related to the human notch 1 gene, its known links to rare diseases, population genomics data, as well as a comprehensive table that shows information collected from major model organism databases. For example, a short text describing the gene function in the table that lists known rare disease association is provided by OMIM, which is run by Ada Hamosh's group, as you heard earlier. If you scroll down, you'll see a table that lists human gene and model organism orthologs in mouse, rat, zebrafish, Drosophila, C. elegans, budding, and fish anise, and their known expression patterns and their gene ontology terms that are based on direct experimental evidence performed at each species. The data that we display here are extracted from model organism databases such as MGI, RGD, Zphene, Flybase, Wormbase, SGD, and PalmBase. And hyperlinks to additional resources such as IMPC and PubMed are also provided here. Finally, Marvel also provides multi-species protein alignment with highlighting feature that allows user to quickly determine whether a specific amino acid of interest is evolutionary conserved or not. Since the publication of the original Marvel paper in the American Journal of Human Genetics in 2017, we published 2 tutorial articles and several YouTube videos describing how to use Marvel. We are continuing to develop Marvel by adding new data set and improving how the data are displayed. If you go to the top page of marvel.org website, you'll see a hyperlink that says, please come try out our Marvel 2 beta. This link would take you to our beta version of the Marvel 2.0 website that has improved user interface, more graphic display, more data sets, and some new features, including batch searches. We appreciate any feedback on this from a user perspective from the field. So after the search, if you figure out that your gene of interest has already been linked to a rare disease, you can try to identify and contact a clinician who are following patients with variants in this gene based on publications. Also, you can search the internet to see if there are any patient organizations that support research on the specific gene. For example, you can go to the website of NORD, the National Organization for Rare Disorders, perform a search based on the human gene name, and you'll get information about clinical resources related to disease. They will also provide links to patient organization groups if there are any. If you have ways you think your research can contribute to developing therapeutics for these diseases, you may be able to find clinicians and support groups who will be interested in learning more about your research. If the gene of interest is not yet linked to a rare disease, you will need to identify clinicians who are following patients with an undiagnosed condition in which your gene of interest is a prime candidate. As discussed in Phil Hieter's talk, if you run your lab in Canada, Japan, Europe, or Australia, you can register in one of these RDMM networks to match with clinicians with undiagnosed patients. These networks are called RDMM in Canada, JRDMM in Japan, which is part of a larger research initiative called IRAD in the country, SolveRD in the European Union, and the Australian Functional Genomics Network in Australia. Note that there are some local RDMM networks that are being developed, for example, at University of Wisconsin and Monash University in Australia, and the number of these local RDMM networks may increase over time. Another way to identify potential collaborators is by taking advantage of public databases related to rare disease research. Decipher in the UK and Geno2MP in the University of Washington have released information related to tens of thousands of rare disease cases that have been genotyped. Anyone can search these databases to obtain basic genotype and phenotype information in these cohorts. By performing a search based on your gene of interest, obviously the human gene name, you can get a list of rare variants that are found in this gene. Each data entry would list a specific variant identified in the patient, together with basic information on their disease phenotypes. Both databases allow people to attempt to reach out to the clinician who have seen these patients, which can be utilized to identify potential collaborators. Finally, there are a number of matchmaking websites that are primarily used by clinicians to connect with other clinicians to identify patients with similar genotypes and phenotypes. One such website is GMatcher, which was already introduced by Aya in her talk. Model organism researchers can register as a researcher category in GMatcher and submit your genes with expertise to connect with clinicians. GMatcher is further connected with many other matchmaking websites that make the larger matchmaker exchange network. This network contains information from over 90,000 rare disease patients from all over the world, allowing clinicians and researchers to identify collaborators on a global scale. We have had many cases in which connections made through GMatcher had led to productive collaborations and publications. I would like to note that there are a number of things one needs to consider before using clinical matchmaking websites. First, your gene of interest may not be the true cause of the disease since it is still considered as a candidate. Second, most users of these websites are expecting to get connected with another clinician and not with the model organism researcher. So they might get surprised if you contact them saying, hey, I'm a Drosophila biologist and want to communicate with you. Third, you may not receive a response from some people you may want to contact since the gene is no longer a good candidate, or simply the person is too busy with clinical duties. Fourth, before reaching out to the clinician, you must think of ways you can contribute to disease diagnosis. One way may be by performing assays to quickly test functions of disease-linked missense alleles in invertebrate model organisms, just like Andy showed you. And another way can be by studying the model organism mutant you have, or those that are available from public repositories like the IMPC, to identify phenotype that match patients' condition in vertebrate model organisms. Fifth, some clinicians may have very tight timeline to publish the first paper because this field is very competitive. Lastly, you must start to think about how your research could potentially help developing therapeutics for these patients and what you want to achieve by directly collaborating with the clinicians. To further facilitate collaborations and accelerate matchmaking between basic scientists, including model organism researchers with clinicians, we are currently developing a global matchmaking platform called Model Matcher in collaboration with Paul Pavlidis and Phil Hieter's group at the University of British Columbia and Zhandan Liu's group at Baylor. One of the motivations to develop this is to complement the lack of an RDMM-like network in some countries, including the United States. We hope to release the beta version of this service within the next couple of months, and information on the progress of this project will be posted on the modelmatcher.net website. Finally, it is important to keep in mind that clinicians and basic scientists may look as if they live in a different world. The two groups have very different cultures and knowledge, and it may even sound like they use very different languages due to the medical and scientific jargons. The RDMM has scientific and clinical advisory committees that oversee matches made between the scientist and clinician through their system to help navigate their communication. However, for matches made outside of this RDMM type network, each scientist and clinician needs to be able to evaluate the integrity of the other party. They need to find effective communication strategy and come up with a series of agreements before any experiment is initiated. In collaboration with leadership teams of large rare disease research consortia and inputs from the community, we would like to establish a set of guidelines, recommendations, and best practices so that collaborations between scientists and clinicians will happen smoothly. Constant collaborations, mutual understanding, and a strong trust relationship will be critical to establish collaborations that are productive and long-lasting. With that, I would like to thank all the speakers of the session for providing valuable insights. I would also like to thank the people involved in the Marvel and Model Matcher projects. And I'll be happy to take any questions if there is time, but I think we should do that on the chat as well. So thank you. I'm just going to Yeah, I'm just going to be leaving this window on. People can take a screenshot if they want to, and these are kind of some of the database that I talked about and their URLs.
I think one of the questions that we're seeing pop up are, can anybody apply to any of these RDMMs in any other country and get a hold of, or at least be part of their registry?
Yeah, maybe Phil may be a better position to answer this, but because the RDMM networks are primarily built to fund research, they can only provide funds that funds cannot cross the border, so that they can only fund projects that are with the two scientists that are in their country. So, for example, I don't think a researcher in the US can register. I did make an account of like the Canadian RDMM, and my information is there, but I don't know if they're going to be matching me with anybody. And again, maybe Phil can.
Yeah, I can. I mean, obviously the budget that we have for CIHR funded, it can only be spent in Canada. So the seed grants go to the model organism researcher, and it's modest amount of money, but the power is really in making the connection, obviously. But we can't fund connections outside of Canada. That's why emerging now is this broader network of networks, like we're trying to really get established where there's cross RDMM interaction. So if there's not a match within Canada, we can look for a match in Europe or Australia. And if we can get this going, then the idea would be that a Canadian clinician scientist could match to, say, a European model organism researcher. That researcher would be funded within the European budget. So the more of these that we have, the better. I saw several questions about, is there a US-based RDMM in the making? And there has been expressed interest, but I don't know of a formal grant yet or a funded project. Shinya mentioned the regional network within the University of Wisconsin. So we have a couple of examples of that. Any region can establish an RDMM if they have a funding pocket, amount of money. So they basically can recruit their local model organism researchers and clinician scientists to try to make connections within the local network. But then that allows, for example, Canadian clinician scientists to our registry to search the regional network, say at the University of Wisconsin, to potentially make a link between a Canadian clinician and a University of Wisconsin researcher that could establish a connection and be funded. So the more of the numbers, it's a numbers game. The more of these that there are, obviously the higher the frequency of the connections that will be made.
And maybe one more thing to add is that the model matcher that we're actually developing together with Phil's group, we're actually using the same software that the RDMM team developed. So if the idea is that if you are in the US, well, the US doesn't have an RDMM, but you can actually register in model matcher, and that would allow you to connect with clinicians in Canada and other countries. Although the problem would be that there wouldn't be any funding, that 25K funding is not going to be provided, but there are a lot of people, and especially scientists, who says that, oh, regardless of the grant, I want to actually work on it, because if I can get some preliminary data, I can apply for an R1. All right, so I think this concludes the session, and so I'll ask the speakers to still hang around until the end of until we actually shut off. So if you still have any questions, please type your questions in the Q&A. If there's some that are not answered, we'll try to get back. If it is from an anonymous attendee, we can't do it. But if you have your email address put in, we'll try to contact you and try to get the answers back to you as soon as possible. Okay, so thank you so much, and I hope this was useful. So if the speakers can still hang on and to just keep an eye on the Q&A box, that would be great. So thank you all, and enjoy the rest of your afternoon and the meeting.
Thanks, Shinya.
Thank you, Phil. Thank you all.